Welcome back to our Brain Talks podcast, a series of 12 podcasts looking ahead to the EBC's Brain Innovation Days in October this year. I'm Sam Polly, and throughout this series, I'll be talking to some of the most exciting innovators in the brain research area as we examine the brain in the life course, from paediatric through to adolescents and young adults, and on to later life. To help me on this journey, each episode I'll be joined by a guest co-host from the European Brain Council. And today we'll be looking at pioneering work that's recently been recognised by the European Paediatric Neurology Society and been awarded an EPNS Research Prize. Beta-propeller protein-associated neurodegeneration, commonly known as Bpan, is a rare, progressive and life-limiting neurological disorder, beginning in infancy or early childhood. Dr Apostolos Papandreou from University College London and the Evelina Children's Hospital is leading research into treatments for the disorder, which could also have positive implications on more common disorders as well. Now, here to tell us more, I'm delighted to welcome not only Dr Apostolos Papandreou, but also Dr Jean-Pierre Lin, a board member of the EPNS and also president of the British Paediatric Neurology Association. And joining us also in the discussion, I'm delighted to welcome Frédéric Destrebec, Executive Director of the European Brain Council. A very warm welcome to you all. Apostolos, before we look more closely at Bpan itself, can you tell us what is the current situation in terms of curative treatments for neurological conditions?
Thank you for your question. In short, there is a almost complete lack of curative treatments for these conditions. Unfortunately, our efforts currently concentrate on decelerating disease progression and alleviating symptoms that our patients have. We have made progress with advanced therapies, such as, for example, deep brain stimulation that Jean-Pierre Lin can talk to us about. But unfortunately, we haven't really progressed as far as we would have liked so far in treating the underlying molecular problem in each patient.
Yeah, Jean-Pierre, I'm turning to you along the lines of what Apostolos just said. What progress can we identify in terms of actual improvement into patients' quality of life and patient outcomes?
Well, Apostolos was saying that is a physiological approach to targeted brain activity modulation. Now, by physiological, I mean it affects the interconnected systems within the brain that are carefully calibrated, in the phylogeny of vertebrate animals and in the development of a child, that have gone wrong in some as yet incompletely understood way. But let's say that the processes are out of focus so that it's very difficult for the system to function. And the systems that respond best to deep brain stimulation are in fact genetically derived movement disorders. But all the genetic disorders vary from each other because some of them are progressive and have a degenerative component, so that the brain continues to be dysfunctional despite the stimulation, and eventually the degeneration wins. Many other genetic disorders have a more, let's say, less progressive condition. They all change with childhood, and this is one of the difficulties. But they allow us to countermand the maladaptation within the brain and allow the true developmental nature to come through. And so many children, particularly if we catch them early, can have a better chance of a more normal development. And normal development means normal growth, normal interactions, communication, attainment of self-efficacy and independence, and so on. So what I think deep brain stimulation tells us is that the nervous system is amenable to alteration. And so long as the deep brain stimulation works, we can sustain that. We know that if the DBS goes off due to failure to charge the battery or due to a technical hardware fault, then everything that has happened in the past in terms of the pathology returns. It's very frightening. Sometimes it comes back with a vengeance because it's become DBS dependent. It's a stern lesson for everyone, but nevertheless, when you switch the system on again, it's extraordinary how you can then return back to the benefits of the deep brain stimulation.
Jean-Pierre, sorry, go ahead.
Yes, so one of the things that DBS has done is it has informed the scientific community that it is possible to at least temporarily reverse or reduce the severity of a condition. And what the scientific community is doing now is looking, well, what is a more definitive approach to changing that maladaptive physiological state? In other words, for those genetic disorders, how can we use gene therapy to give a more complete, a more definitive, once and for all solution in typically a developing child rather than a fully grown adult in their 30s and 40s. So I think the DBS has been if you like, a herald of the more precise advanced therapies to come.
And we'll ask Apostolos about that in just a moment, about the precision medicine to come. But just can you give us just a quick idea of how innovative and rare is the DBS itself? How widely used is that?
DBS has become, I would say, standard therapy in most developed countries, but by far not evenly available or distributed. It requires a great combination of team players and a multidisciplinary team to actually derive the benefit from DBS. Unfortunately, the model of dystonic child meets surgeon who implants a device and then is sent home, it just won't work because it requires adjustments, troubleshooting. It's very important when you start off a process like deep brain stimulation that you have goal setting and that you are able to absolutely understand parental expectations and at the same time give some indication of what to expect in the first months and years. Because it's electricity, people anticipate that you switch on the light comes on. But this is the brain we're talking about. And I have a knitting analogy about the brain, which is that as we grow and develop, our brain knits up into patterns of function. As the brain descends into chaos, then that pattern is disturbed. The disturbed pattern then causes the brain to knit a disturbed brain pattern. So you get a continuously imprinted maladaptive process on the brain. What the DBS begins to do is to allow some more functional postures and movements to populate that pattern. And then you then gradually keep changing that pattern until you have a more perfect, as close to developmentally normal pattern.
Jean-Pierre, earlier you touched on the future of personalised medicine. Apostolos, how important a role is personalised medicine now playing, and what innovations are you using?
I think it has a huge role to play in pediatric neurology and in human disease in general, dare I say. We, as Jean-Pierre said, we have advanced with our therapies, but we really need to go down to the very basis of the issue. We need to go down to the molecular basis of the problem in each condition, and especially for neurological conditions, and address that in order to have good outcomes and outcomes that are the outcomes that our patients deserve. So, for example, in neurological disorders and in Bpan, for example, there is a there is an innate cellular mechanism of self recycling and self renewal that's really important for brain cells, because brain cells, they do not have the capacity to divide and regenerate. So that they need to have really robust waste recycling and disposal systems. And in BPAN, this system is dysfunctional. And unfortunately, it affects the whole brain. It affects the areas of the brain that control movement, but also the areas of the brain that control thought processes and development and so on. So it's a pan-brain disorder. And in order to treat that, as I said, DBS might work in controlling the movements for a bit, but it won't work for the whole brain. So you're looking at innovations that you talked about. Innovations are, first of all, understanding the molecular mechanisms of these conditions. And we can do that now because we have technologies by which we can study human-derived tissues, and I'll explain that in a moment. But we can also use models to treat for disease-specific treatments, to search for disease-specific treatments, and then apply those in the preclinical and clinical settings. So I'll be more specific. We have a technology called induced pluripotent stem cells that we're using right now. Induced pluripotent stem cells can be derived from any type of patient tissue, for example, skin fibroblasts or blood cells. And these cells are, in a way, manipulated so that they become stem cells. And by that I mean cells that can then be subsequently differentiated into any type of cell in the body that you want to study. In the BPAN case, it's dopaminergic neurons. It's a specific subtype of neurons that's particularly affected in BPAN. And when you have induced pluripotent stem cells and then the cellular subtype of interest, you can study the disease in a pathophysiologically relevant model, which you can use to understand what causes disease in the first place. But as I said, you can also use this model downstream to, as we did in our research, to treat these cells with thousands of drugs to find drugs that correct these defective processes of waste disposal and recycling. And once you've identified those drugs, you can then think about translating them to the patient as soon as possible.
But because you touched on the human-derived tissue, historically there's been a lack of that then. That's been a real hurdle to overcome.
Absolutely. And that's, as you say, historically one of the major hurdles in neurological disease research. We haven't really had the chance to study the human brain, apart from sort of post-mortem studies and so on, which are not as informative. And also, we've studied other preclinical models, animals, mice, for example, or larger primates, which can give you a lot of information, but often not as adequate as when studying human tissue. So I'm not saying that mice or other animal models are not useful. In fact, every model is complementary, and we might have to use other preclinical animal models in BPAN before clinical translation to the patient. But the induced pluripotent stem cells have given us a tool that we haven't really had in our asset before.
So Jean-Pierre, and we'll look a little more closely now, I think, at Bpan. You've talked about it a little already, and the work that's being done in the S area. But just very quickly, for anyone that's listening, it is a very rare disorder. Just in a sentence or two, Jean-Pierre, can you just explain what Bpan is and how it manifests itself?
Well, it's very interesting because Bpan eluded the radar for quite some time because of the way it presents in early infancy, and because of the way that our minds were conditioned in relation to other causes of neurodegeneration with brain iron accumulation. So the classic Bpan will present in infancy with a child who maybe has a slightly small head, has developmental difficulties, may have very difficult to treat and manage seizures. And the initial scans that are performed really are blameless and offer no clue. Now, the difficulty is that changes in the brain occur after about the age of 10, which means that you have to have the bright idea of rescanning in the second decade. Now, Bpan has another name in the adult literature called SENDER. I've just temporarily forgotten what the SENDER acronym stands for, but in the SENDER population, the changes on the scan are visible, and this is, of course, the iron accumulation. The iron accumulation occurs in the globus pallidus, but very, very especially it occurs around the it occurs in the brainstem around the dopaminergic center, the substantia nigra, and there's a halo, if you like, around that. So it's much, much easier to diagnose a condition when you have a radiological signature tube. So most of us have discovered our Bpan cases retrospectively through chance images. For instance, somebody gets a scan after an emergency admission, and then the scan has the particular signs. But of course, knowing now that the radiological signs change, people are in advance. And one of the conditions which Bpan can resemble or be a pseudo-mimic is Rett syndrome. So Rett syndrome is quite common. It's well recognized. There are many different genetic causes for Rett syndrome, but Bpan is Rett syndrome-like.
And it's very rare, Bpan, isn't it? How rare is it?
It is rare. Sorry for talking over you. It is rare. It can present later on in life. So some Bpans can present in the second decade. And obviously, for those cases, they may have a better chance of neurodevelopmental progress, be ambulant, be able to talk. And when they start experiencing symptoms, it's as if there's a slowing down, and they have a more Parkinsonian presentation. In other words, they would be seen to have an early onset Parkinson type of cluster of symptoms. So the age at which the condition expresses itself both imprints the severity on the disease, and the rule is early onset, worse prognosis, later onset, better prognosis. But also the types of symptoms are different. It's as if it's borrowing its symptom profile from another rule book.
Apostolos, we mentioned a few innovative techniques at the beginning of this talk. Can you maybe say a word about how these were used in your research on Bpan specifically, and what have you found thanks to these?
Sure. Thanks, Fred. Effectively, what we did is we wanted to study tissue of interest or cells of interest. As Jean-Pierre said, we wanted to study cells that richly populated the substantia nigra, and these are dopaminergic neurons. We wanted to do so because there's a profound iron accumulation in the substantia nigra later on in life, and the substantia nigra is the most affected area. but also because Parkinsonian features are one of the most prominent disease features later on. So what we did is we used induced pluripotent stem cells that were derived from patient fibroblasts and age-matched controlled fibroblasts. We then differentiated these into dopaminergic neurons, and we studied this brain model in the dish, so to speak, in order to identify how the patient cells behave differently in the dish compared to the control brain cells, and hence what leads to dysfunction and early death, neurodegeneration. And once we identified the defective mechanisms, we used more innovative techniques. I'll talk a little bit about high content imaging and high content screening. Effectively, we tested thousands of drugs for their capacity to restore the defective phenotypes or the defective cellular functions that we identified in the patient. And we used that, and we did that using a powerful sort of high content, meaning a microscope that was able to capture many images in a very short time, and test many conditions and many treatments in a very short time that would be impossible 10, 20 years ago. Induced pluripotent stem cell technology combined with high content screening were used. And what we found is we found several compounds, small molecules or drugs, if you will, that we can confidently say are very efficient in correcting the defective waste disposal and recycling system that we see in the patient cells in Bpan. These also correct other defective functions that come as a result of the primary problem, which is the defect in the waste recycling. And hence, we now have compounds that have a real potential to be of therapeutic benefit for our patients. We need to do a lot more work. As I mentioned before, we might need to do more work in other preclinical models, such as a mouse or and so on. But we have the potential to provide meaningful disease-specific and effective novel therapeutics for these patients. And the other interesting bit is that because Sam mentioned this briefly in her introduction, this waste recycling and disposal system called autophagy, which is Greek for self-eating, it's very important for neurons. It's very important for brain cells that do not have the capacity to regenerate after injury, for example. So we understand now that this process is very crucial, really, for neuronal homeostasis, and we see it increasingly implicated in the pathophysiology of very common conditions such as Parkinson's and Alzheimer's disease. So by understanding Bpan and its defective waste recycling process, waste disposal and recycling process, and by understanding what we can use to treat Bpan, might give us insight into more common conditions such as Parkinson's. So that's also one benefit that might come out of this work to the wider community.
And Jean-Pierre, do you have anything to add? Apostolos also talked about more work to be done in terms of translating clinical translation and therapeutics. Is there any other work, other steps that we need to cover that you'd like to add?
Well, I think the models that Apostolos was describing that allow rapid screening of potential small molecules that could be helpful. The question, I think, might arise, you have something that works in a laboratory, in the whole organism, it may be a different prospect. So you mentioned the mice. But then the question is, how old must the mice be? What developmental stage should they be at? When do you offer them at? When do you offer this molecule that can do something different? My instinct would say early on in the development of the organism. And then, of course, the issue about early delivery, with these simple molecules, shall we call them, it still requires early detection that is going to get this condition. So in other disorders where the advanced therapy has become, shall we say, standard, it is not the perfect way to manage things, because it's been determined that if you're born earlier and you get management even earlier, then you have a better effect. So the question then is, in utero management, if you like. So there's always a regress about how to deliver something, and in the initial stages, the cases are in a terrible state. Something must be done. It's a humanitarian actually halt things, but then in a very short order, you want to roll it back to the earliest stage as possible. And the muscular atrophy will show a picture of another later being pushed around in a pushchair by a young who was treated earlier, and the younger sibling has got full ambulation. The older sibling is alive, but not able to walk. I think timing becomes a crucial question once you have something that has a possibility of being useful.
Fred, let me just bring Apostolos. You wanted to say something?
Just very briefly to say that absolutely we need to diagnose these kids as early as possible, because early intervention is key. But apart from that, we also need to really understand the natural history of these conditions, because you've given them the treatment. How do you know that it's worked? And because there's obviously variability in the clinical presentation, and there's a big spectrum that's getting larger and larger. So you really need to understand not only the natural history, but also, dare I say, you need to think about developing clinical, radiological, but also biological markers by which to not only diagnose, but monitor a disease severity and response to therapeutic interventions. So in order to go to trial readiness, you don't only need a small molecule or gene therapy and then a mouse and more preclinical work, but you also need these things happening in parallel, the natural history study and the biomarker discovery. And imagine how difficult this is in an ultra rare disorder.
Let me bring Fred in. Fred, what are your thoughts on what you've heard so far? And I think you wanted to ask Apostolos about some of the challenges as well.
Yeah, sorry. No, indeed. And I think that what we've been hearing so far from Jean-Pierre and Apostolos is quite striking, as we are really at the, I should say, collision of multiple challenges. First and foremost, the inherent complexity of the brain, of brain research, of understanding mechanism of specific conditions, particularly here in the context of a rare disorder. But also, it seems to me, coupled with the challenge of conducting research in pediatric or for pediatric patient groups. And here, maybe Apostolos, you could tell us more as to how you're going about, let's say, combining these different facets and how you have been able to overcome that.
Sure. Well, first of all, I have to say that our patients and our families are just phenomenal in their willingness to participate. And they really are driving this work, and we couldn't have done anything without them. And whenever you ask anything of them, they're always so willing. It's really amazing, and they are the ones that motivate us to continue this work. The other obvious thing is to collaborate with people not only within the country, but within the continent and across the globe, because you have to in an ultra rare disorder, and one lab or one institute can only do so much, whereas with for example, some clinicians in Oregon are currently doing a natural history study. They've already recruited 168 children. Other researchers in Germany have created a mouse model. There's another two mouse models of Bpan in China. Already published. There's Drosophila models of Bpan. All these are helping to understand disease pathophysiology, but provide models on which to further test our novel therapies. So we need to collaborate in order to overcome the difficulties and the challenges. One of the main challenges I probably have alluded to is the ultra rareness of this disorder, and how do you motivate stakeholders such as the National Health Service to fund such an expensive research for 50 children, for example, in the UK, which might require millions and millions of pounds with no guaranteed outcome, and with a lack of natural history studies and with a lack of biomarkers. So how do you do that? And dare I say, how do you involve industry partners in this, who have a lot more resources than academia? Jean-Pierre, I don't know whether you want to continue in this.
Well, I want to first of all, I want to echo the power of the families to generate the energy and the of undertaking these things. I actually remember when the European Leukodystrophy Association, ELA anyway, set up, and a series of families all had children and young people with leukodystrophies. Their first international meeting in Paris attracted researchers from all over the world, Texas, China. It was an exciting meeting because here was a commitment from these major institutions to invest in rare disease. So individually rare, collectively important. So I think grouping together, as a series of kindred but separate and different projects helps to, I think, pool resources that are common to them all. The natural history, I think, is an absolutely crucial thing. And most who've had an interest, say, in movement disorders and so on, have got cohorts. And they maintain their cohorts, through thick and through thin, because they know that if anything can change for better or for worse, the cohort will measure that, and then it can be used as a backcloth. Deep brain stimulation depends on cohorts. I was just commenting on to some colleagues how many of our graduates have gone to university. It is a success when people leave childhood, go to university, higher education, do software engineering and other amazing things. That's a marker of important change. And we don't really have enough of that information beyond, say, the mid-teens at the most. And certainly adult services are not configured to collect that data. But, what happens in the third decade is important. So establishing, these networked cohorts would, I think, be an important resource against which to judge the benefits of something which is introduced early on in life, and also would allow health services worldwide to understand, well, if we are producing, more complete young people at the end of the process, less dependent, more satisfied with their lives, then that is an achievement. Having said that, I've just had a conversation with a young man of 20 who, from my perspective, is doing amazingly well, but from his perspective, he's still not doing enough. He plays golf. He can independently take a group of friends to watch a Liverpool Cup final at Wembley. He's driving a car. He was learning to drive last May. He got his license. He's doing all this, living independently, but not perfectly happy. And I think this is another issue that, happiness, contentment, satisfaction, it's not human.
Yeah. And different expectations from different patients, I imagine, as well.
Want something more, something, that is just out of reach. Otherwise, if we've achieved all that, we may as well just give up.
Fred, let me just bring you in before I wrap up. I'd like a final thought from you all, but did you have anything to respond to Apostolos or Jean-Pierre on the challenges and further needs they were talking about?
Well, actually, I had a follow-up question to something I think, Apostolos, you mentioned earlier, is the potential for translation into other more mainstream disease areas, such as, I think you mentioned PD, and I guess other motor disorders. Is that potential for translation not, let's say, a solution in itself in terms of developing further the research that you're conducting?
Absolutely. I think it's a very good question. You're right in that, for example, our understanding and treatment options for Parkinson's disease over the last 20 years have largely come from studying rare monogenic conditions such as PINK1 and parking and alpha-synuclein, and you see mitophagy regulators being developed for Parkinson's disease and so on. So this is really of interest to the wider neurology community and the wider industry community, and we could possibly utilize this to attract more interest in our studies and in our research, and possibly attract more funding. I dare say that Bpan is probably already exploiting this in the nicest possible way. Other similar brain iron accumulation conditions who have been whose molecular basis has been known for 20 years rather, they haven't got as many research labs around the world working on them, so devotedly. That's possibly because autophagy and Parkinson's disease, they're quite popular at the moment, and people are very interested in researching, and funders are more interested in funding relevant projects. So absolutely, I think you're right. We need to really ride the wave, if you like.
We've nearly out of time, but before we finish, let me just wrap up with all of you, because I'd be interested in a final thought from you all. You've spoken about the successes that you've had and the potential wider implications as well. What are your hopes for the future? And that could be about using these innovative techniques, or perhaps how hopeful patients and their families could be. Are we even looking for a cure? Jean-Pierre, let me come to you, and then I'll come to you, Fred, for how hopeful this leaves you, having listened into this podcast.
Yes, at the moment, I always have to tell families that we have management strategies. We don't have any cures. DBS is a management. It's not a cure. And they always find that quite difficult. I think our health services talk about go to your doctor to get treatment. But mostly a palliation of what is happening to you or to your child. And that's something which does set people up for disappointment. They came for a treatment, they get a management, the condition is still there. So I think with the new developments, we are trying to turn that leaf and change over from management, palliation, to something which means that, yes, life can go on, but without the shadow of that condition.
Fred, and then I'll finish with you, Apostolos.
Well, yeah, thank you, Sam. Actually, there was one thing that came to my mind, is the fact that obviously, we have identified, let's say, common features that we have spoken about in many of our activities at EBC in the context of the Brain Innovation Days, in some other of our podcasts. But what I realized today is particularly speaking of a very rare pediatric condition, there is one thing that can make us all hopeful, and particularly patients, is the commitment and dedication of Apostolos and Jean-Pierre and their colleagues in continuing their research, in continuing their work, and striving really to try and offer solutions. Obviously, today in the management of the commission, the condition, but hopefully tomorrow for proper disease-modifying therapy.
And Apostolos, let me finish with you. How hopeful are you for the future?
I have to be optimistic, but also being realistic, as Jean-Pierre and you all said, these things do take time. We are hoping that this comes tomorrow, but realistically, it will take time. And but I want to reassure our patients that we're really doing our utmost to bring these novel, meaningful treatments to them, as soon as feasibly possible.
Well, it's been really fascinating to hear from all of you today, and Apostolos and Jean-Pierre, to hear about the work. That's all that we've got time for today. So thank you all very much for joining us here on the Brain Talks podcasts. And thank you everyone also for listening or watching in. Do join us again soon for another podcast as we look ahead to the Brain Innovation Days and continue to explore the brain in the life course. Until then, bye-bye.